So please allow me to introduce you, Arvind. Thank you so much for your time. So
Thank
friends,
you.
welcome to this Bioclues and IEEE Computer Science initiated webinar on linking tumor-derived phenotype information with genetics for personalized medicine via HTS by Arvind Rao, who is a professor at the University of Michigan in the States. And I have before me a very prolific account of his, a wonderful profile, which I'll probably narrate down in just a few minutes. Arvind is a professor in the Department of Computational Medicine and Bioinformatics at the University of Michigan. His group uses image analysis and machine learning methods to link image-derived phenotypes with genetic data across biological scale, including single-cell, tissue, and radiology data. Such methods have found applications in radiogenomics, drug repurposing based on phenotypic screens, and spatial profiling in tissues as well as in spatial transcriptomics. Arvind received his PhD in electric engineering and bioinformatics from the University of Michigan, specializing in transcriptional genomics, and was a lane postdoctoral fellow at the Carnegie Mellon University, specializing in bioimage informatics. Arvind was introduced by us to Sri H.R. Mohan sir, who has been kind enough, and he always introduces wonderful personalities. And today brings us a wonderful limelight Thursday, and the floor is yours, Arvind. Thank you so much for your time.
Thank you so much, Prash. I really appreciate it. Thank you again to Mr. Mohan for introducing me to this group. I apologize for the trouble it took to get started, but hopefully I'll be able to make up the time as we go through. So very broadly, I wanted to get right into it, and I think given that this is an intersection of Bioclues, IEEE audiences, I thought it would be good to talk about things at a fairly high level. And of course, if questions come up that are deeper, then we can definitely revisit them towards the Q&A. But I just wanted to give you a broad flavor of what we are doing at the intersection of electrical engineering, computer science, and biology slash cancer medicine. So I think the biggest fact about AI in the past 1 year has been that last year we had two Nobel Prizes for AI in science, one in physics and one in chemistry. And this is the physics one for the Hopfield neural networks, and the chemistry one is for discovering protein structure. So very quickly, AI has become mainstream in a variety of biological applications. And based on this sort of energy, there's in fact a very popular challenge called the Nobel Turing Challenge. This Nobel Turing Challenge hypothesizes that by 2030, a completely automated engine will make a Nobel Prize winning discovery. Right? So Nobel Turing is this a combination of Nobel, who is the Nobel Prize person, and Alan Turing, who you might have heard of in the computing circles. And the idea being that with all that's happening in AI, the possibility of an AI-based engine making a Nobel Prize-winning discovery is distinctly feasible by the year 2030. So this puts us in a very interesting place where we have lots of conversations happening in parallel. With Nobel Prizes becoming, something for AI to work with, obviously there's a lot of enthusiasm in the community. Last year was the year of AI in science. This year, 2025, is the year of multi-agentic workflows in research. There are aspects of AI ethics people want to talk about. People want to talk about human AI collaboration. You want to talk about AI co-scientists. All these kinds of terms are becoming a lot more mainstream. And so this behooves us to kind of understand where can these engines be used and how does one think about productivity in this new era where AI is now going to be not just an action partner, but also perhaps a thought partner with scientists. So as Prash said in the beginning, my group's work is on image informatics. We do a lot of work in looking at images as data and analyzing them, extracting all kinds of information, which could be from cell information or tissue information or even radiology information. So that's something that we do quite a bit of. Um, right? So this is cells, this is tissue, and this is radiology images. But I'm going to focus in the beginning of the talk on cell images. And HTS is essentially high throughput, high content screening, and it's a very popular modality of data acquisition for drug discovery. So this notion of trying to discover drugs or to repurpose drugs based on phenotypic aspects of cells morphology is what is called phenotype guided drug discovery. It's a very popular way of doing things. And as you can imagine, with the robotization of science and all these aspects of acquiring large amount of data, the potential for machine learning and AI couldn't have been bigger, right? So how does that all come together? I'll show you one case study. So we are working with a disease called triple negative breast cancer, also called TNBC. And the idea there is to do a high throughput screening experiment of the following kind. We have these experimental plates that are about 96 well plates. We can seed the cells into different wells, and each well receives a distinct perturbation. It could be a drug, it could be a gene knockdown through CRISPR or RNA interference, what have you. But the bottom line is now every well is its own experiment, and from every well you can now take a picture of the morphology that arises as a function of that perturbation. So you can now do these perturbations at extremely high scale, and what you're getting now are images that are readouts of that sort of experimental process, right? Just a very quick word. These wells, when you zoom in and look at the EM pictures, electron micrograph pictures of these plates, these look a little bit like this. It's a very unique kind of gridding structure in the bottom of the plate, and it will become relevant in a few slides in the sense that when you shine light to take a picture, these griddings that you're seeing, they diffract light in some unpredictable ways, and so your data can be quite noisy. We are essentially, kind of telling you about the kind of pre-processing we have to do before we can actually do data mining. So now how this data looks like is the following. There's 40 plates worth of perturbations we are doing. Each plate is a 96 well format. What you see here in color, not the blue color, is controls, positive, negative controls. The blue colored wells are the actual experimental perturbations. And what you're doing is you're now trying to screen for the entire kinome, all the kinase genes, and trying to perturb them one by one and ask the question, how does knocking down a particular kinase affect the morphology of these triple negative breast cancer cells? Now, I keep saying morphology. What do I mean? So I wanted to give you some intuition here. The typical morphology of these triple negative breast cancer cells is this protruding finger-like structures called the mesenchymal cell type. Okay? And if you look at them in actual images, this is how it looks like. You have these long projections that indicate how these cells are innervating their microenvironment. And you can imagine why it's a very aggressive disease is because it can move very easily, and the mesenchymal cells can now transport essentially the disease phenotype wherever the blood takes them, right? So they're very easy to move, extremely mobile, right? However, the point of this experiment is the following. We want to identify those kinases that, when knocked down, will change the phenotype from being long projections to becoming no projections, right? So essentially, there's recalcitrance of this protrusion, and instead of being mesenchymal, it becomes epithelial. And epithelial phenotype is a lot harder to move. It doesn't move as easily because there is no amoeboid movement possible. And so you want to essentially change the morphology of these cells through a drug or through some kinase level perturbation that will prevent these cells from moving. And if they don't move, guess what's going to happen? They'll stay localized. You can hit them with radiation, chemo, whatever it is, and you can clear out the disease a lot more favorably than if they were much more mobile. So this is the intuition of what we are trying to do. And what I said also is that you can do these perturbations on every well, and for every well you can take about 6 pictures with a high content microscope. So in the end, with 40 plates worth of data, all the positive, negative controls, roughly you have about 20,000 images. Okay, so that's how much data you have to mine to be able to find these hits, right? And so, in the quest of trying to do that, there's a typical pipeline one could follow, which is, I guess, a consistent pipeline for any kind of biomedical data. There's always some pre-processing involved, be it genomics, be it imaging. You get some processed data as a function of the pre-processing. You extract features of different kinds. You then use those features to do data mining or whatever kind of machine learning approach you want to be able to use. Use that to be able to score your data, find the hits, hopefully. And then once you find the hits, you can try to map them to canonical pathways to understand what's the biology that's underlying those hits. That's the big picture. Now let's get into the specifics, right? And like I said, I'm going to zip through a lot in the interest of time, but I'm hoping to keep the intuition there. So if something is not clear intuitively, please feel free to interject and ask. So the first step is pre-processing. You have this data, and you can see, if you look, just stare a bit at these pictures, you will see that there is actually a gradient effect that happens due to the scattering of the light by that fine gridding that I had shown you a few slides ago. So the images are not as clean as you would like. There's a diffraction artifact. And so we clean that up. We do some pre-processing to segment the foreground from the background. Then we segment out these cells. And what you're seeing in red are essentially the areas where the cells are located. Whatever is not in red is stuff in the background. These images are actually directly from the microscope. So what I'm showing you here is that this data is pretty messy, which is, I guess, the reality that we deal with when we're trying to do AI in the real world, right? Because AI, it's always easy to do projects where, you can curate the data and get AI to work for you. But for it to work routinely in routine deployment, you have to be willing to work with the noise in the data. So I want to make a mention of that. Now that you have extracted these blobs, in the ones in red, you can extract features. And in this case, the features are morphological features, because I'm interested in looking at protrusions. That's the morphological feature. So I can measure things like the area, perimeter, form factor, all those kinds of things, and really get a huge cardinality of features, right? And we could have this philosophical discussion about why we choose this approach compared to deep learning. Of course, you can use deep learning in the modern era, but I'll tell you in a little bit why deep learning would not work for this particular problem. It could work for a flavor of this problem, but for this particular problem, it was hard to work, and I'll tell you why. So we do the good old stuff where we simply get features, get a huge feature matrix where you've got, every row is an image and every column is a measurement from that image. Right? So you can get this big data matrix. And now you can feed that to whatever you want to do. And what is the point? I want to be able to build a classifier that will be able to look at an image and tell me whether it is in the the mesenchymal category or in the epithelial category. So typically the cells will be mesenchymal, but we want to look at those images that are in the epithelial category, right? So the ability to call these two morphotypes is key in the design of the classifier. Now comes the big question. We want to build a classifier, but we need labeled instances. So there's 20,000 images, and we want some training data to be able to build a model. So what do? So I went to my clinical colleague who has started this experiment, and I asked him, hey, can you give me some labeled data? And he asked me a very simple question. He said, how much data do you need? I said, I don't know. I don't know how, what the effect size for this particular morphological feature is. So give me 100, 200 images. He said, I don't know how much I'll be able to do, but I will sit with you one afternoon, and then I will label as many images as I can. He's a clinician, right? He sees breast cancer patients. So when he says he's got one afternoon to give me, I've got to accept that and hope that he can give me enough data. And he's a very assiduous, individual. He takes the time to do it right. So he spent a whole afternoon, and in the end, he gave me 33 high quality instances, right? Now, I expected to get 200 and I got 33, right? But these are very high quality annotations. So let's see what we can do. This is one of the reasons why you can't use deep learning, right? You don't have that much data to learn from. And because you have some sense of the physics of the problem, feature engineering is probably your only way to be able to salvage the issue, right? So we did that. With these 33 examples, we built a random forest classifier. You could build you could use anything that you like, my favorite is random forest for a variety of reasons, including the fact that it gives you, cross-validation for free, it does a lot of bootstrapping, all those kinds of things. So I used a random forest classifier to train a model based on these 33 instances, right? And now I can run this model loose on all of the remaining data. Remember, I had 20,000 images, which are my overall data. 33 of those are labeled. I learned the model. Now I need to run that model on the remaining 19,000 whatever, 877, right? 867, right? So what you end up getting is this aspect that you have all this data that you want to be able to munch through, and you can find hits, right? You can find hits, that is, which genes, when knocked down, change the phenotype from the mesenchymal to the epithelial, and you get 351 genes. Now, if you are talking to a clinician, this number is huge. Expecting them to follow up each of these 351 genes in a lab is next to impossible, right? Biologically speaking, doing experiments at this level is really hard. So the first thing he told me was, can you give me a smaller set of genes to work with? It's too much for me to work with. So can you reduce this? And I said, okay, give me more data and I'll try to build a better model. Hopefully it'll be able to discriminate what is truly happening versus what might be happening on the fringes. But that is easier said than done. If I again had him sit with me another afternoon, not only would I feel guilty that I'm taking his time away from his patients, but again, I might just get about 30 instances, which may or may not be as valuable from the overall training perspective. So how do I extract data in a way that is meaningful? This is where ideas from allied fields come in. So you've all seen Amazon and Spotify and Netflix and Facebook, right? What do? When you open Netflix account, it gives you 10 random movies to watch. If you like some of those movies, you give a thumbs up. That means you like the movie. In some other movies, you might not like it, you give it a thumbs down, and the algorithm learns that you don't like them. Over time, it begins giving you more and more movie recommendations that are aligned with your sense of what you like versus you don't like. So the algorithm is learning your preferences based on how you label data as being good versus not good. Same thing with friend requests on LinkedIn or on Facebook, or books that are recommended for you to read by Amazon. All of these algorithms have one thing in common. They do what is called active learning, right? Active learning is a principle where the following is done. You have some initial labeled data. In my case, that was 33 instances. I can build a model. It may not be a great model, but it's some starting model. And I can use that model to label all the remaining 19,000 odd images. Okay. Now, when I use the model to make a prediction on those 19,000 images, there's a probability that I can get from the model, which tells me, I think that you are mesenchymal with probability 0.9, or you are mesenchymal with probability 0.6, right? And so, because you get these probability values accompanying the prediction, you can use the uncertainty in the prediction to only flag those examples that are most confusing to the model. That's how humans learn, right? When you're in a class and you're being taught a concept, you raise your hand and you ask a question. You don't ask questions about things that you know. You ask questions about things that you do not know or you're heavily uncertain about. So that whole idea is called uncertainty sampling. So we do uncertainty sampling with all these test images, and then we query the human expert for annotation on only the images that we find to be most confusing. This gives you a very quick way to be able to extract a lot of information from the human expert without necessarily having them do things de novo, right? So this is an idea that's been out there in other places. We simply co-opted it for our problem. So now in about 2 hours, I went from 33 instances to 133 instances. I got 100 instances in about 2 hours because all you had to do was look at the image that I was asking him about and saying, is this category 1 or category 2? That's all you had to do. And that really sped up the learning process. So we did this a few times, and the second round, you run the same thing again. You now get 256 genes instead of 351 genes. We reduced our search space by 100 genes. You can keep doing this a few times until you converge to a set of genes that you are comfortable with. So now let's ask the question, that's all very nice. What did you finally find? So when we looked at the genes that made the cut after repeated cycles of learning, training, and labeling, we converged on a pathway, I'll come back to this in a second, called the PI3 kinase pathway. Now, I don't know how many of you are in the biology field specifically, but if you're in breast medical oncology, the PI3 kinase pathway is one of the most canonical pathways that is involved in cancer etiology, right? So what did we just say? A completely machine learning based algorithm, which had no biological domain knowledge, just through the process of iterative interplay with a human expert, was able to converge to the role of the PI3 kinase pathway in this terrible disease. This to me represents a very interesting way of trying to bring AI engines and human experts together. Because AI is very good at searching through stuff, and humans are very good at being able to find counter hypotheses and those kinds of things to be able to derive information from the data, right? So now that we found PI3 kinase pathway, let's go back a little bit to the biology of the disease. It's hard to just talk about PI3 kinase pathway without understanding what terrible this disease is. So this is called triple negative breast cancer. It's triple negative because it doesn't express 3 canonical markers: the estrogen receptor, which is called ER, the progesterone receptor, which is called PR, and also doesn't express the HER2 receptor, right? And the reason that these 3 receptors are really important is if any of these are being expressed by the tumor, you could give endocrine or hormone therapy to help the patient. But when all these 3 are negative, there's nothing you can do to help the patient, unfortunately. And it's so aggressive that even by resecting the tumor, it will come back, unfortunately. So we really need a way to be able to deal with this disease using clever approaches. So what did I just tell you? That the PI3 kinase pathway seems to be one of the most important pathways in the progression of this disease. Which means if you were to design a PI3 kinase inhibitor, could that do a good job in arresting the aggressiveness of this disease? And that's exactly what we did. So we used what is called a combinatorial approach, where we took 2 different cell lines. One is called MDA MB231, one is called SUM149. Both of these are breast cancer cell lines. And if you only use, so the drug that was commonly used at the time was called the MEK inhibitor, MEK, MEK inhibitor. So if you this is how the cells grow when there is no drug. They are pretty, you can see the proliferation is high. There are all these projections. You can use the MEK inhibitor, which is this drug, and you see some reduction, but not much. Similarly, when you use the PI3 kinase inhibitor, which came out of our data mining study, again, there is a reduction, but not much. But when you combine the two, you get drastic improvement in the performance of this combination against the disease. And so this led to our clinician colleague wanting to run a clinical trial where he proposed adding the MEK, the PI3 kinase inhibitor, as an additional inhibition beyond the MEK. And that result is still expected, but from what I hear, that was the most promising thing that he had discovered in a long time. So we kind of make a few points here, right? One is that in the case of this particular problem, the physics told us that morphology was important. I've told you about this notion that pre-processing can be really important, and sometimes it's the unsung hero of the process. Otherwise, you have garbage in, garbage out kind of things. And one can use deep learning provided you have data sizes that are much bigger. But because we had a much smaller data set to begin with, of this 33 examples, it was hard to use the deep learning route. So I'll stop there, and I'll now change gears to something else in the next 15 minutes, right? Which is this whole promise of immunotherapy for personalized medicine, right? We all know that, there's chemo, there is radiation therapy, there's surgery, and immune therapy has come up as a very, very big modality for treatment. And this got Nobel Prize a few years ago. And there are tools coming online that let you take a piece of tissue, a biopsy, and stain that tissue for a variety of different markers. So what you end up getting are these amazing pictures. This is actually colorectal cancer. Amazing pictures where every dot you see is one cell. You can get a single cell resolution. And every color of dot that you see is a different type of cell. It could be a breast cancer cell, it could be a fibroblast, it could be an immune cell, whatever. Right? And so you can get these really incredible images. But the big question now is, while getting the data is now not that hard, analyzing it is still a very, very big challenge. So right now, as of 2025, the instruments are becoming affordable. People have these instruments in their labs, in their hospitals, and they're getting this kind of data as we speak. But there's just not enough set of good tools to be able to analyze them. So essentially, it's just collecting digital dust in the hard drives, right? So, and if you come from biology, you think about the fact that we are three-dimensional entities. Biology is three-dimensional, right? We have cured cancer in petri dishes. We have cured, we have destroyed cells that are growing in two-dimensional sheets. But it's this three-dimensional context and the fact that we have, vascularization, you have hypoxia, all these different kinds of things happening that are driving the architecture. And so you need better methods of making sense of this data. So I play a lot with Legos, right? I'm sure several of you have done Legos before. So let's look at this example of a house, right? Built with Legos. If you were doing single cell sequencing or bulk sequencing, you could take this data, get all the Lego bricks, you could sort them, you could arrange them and do all kinds of pretty things. But if you don't know that it was a three-dimensional context, you will always miss the signal, this part of a bigger house. So spatial data is everywhere, and the availability of instruments to acquire that data is also available. The problem is the analysis. So in that regard, we said, this is actually a place where you can use machine learning, deep learning ideas to make sense of this data. And I know this is a very heterogeneous group and there's a lot of computer scientists in the audience. So I'll say that if you're looking for a hard problem, not hard I mean interesting problem to work on, I can't overstate how interesting biology has become for computer scientists, right? There's just a huge amount of data. We don't know what to do with it. And there are a variety of techniques that already exist in computer science that if we could co-opt and adapt into our problem space, we could do amazing things. Okay, so I'll just mention that to you and I'll move on. So a typical image that we are getting looks a little bit like this. Here again, this is actually a piece of lung cancer tissue. And every dot you see is one cell. Every color of dot is a different type of cell. And we get these images and our clinical colleagues say, get me a number, help me make sense of this data, right? And so what we did was we pursued a problem where we wanted to classify images from 6 different kinds of pancreatic disease based on this kind of staining of cells. And while we can stain for a lot of cells, we just take 3 cell types just for illustration. Epithelial cells, which are cancer cells, CTLs, which are immunoactivating cells, and Tregs that are immunosuppressive. Okay, so we take this and we look at these images and say, what can we do? Long story short, you can design a graph-based neural network to be able to classify these images into different categories. And that's not a surprise, right? Frankly speaking, deep learning is supposed to do well in classification tasks. But what can deep learning do that we couldn't have done otherwise? So we inserted what is called a self-attention layer into this graph neural network. And the outcome of that was that we could obtain a heat map on these point patterns that told us which cells are most relevant to discriminate different grades of pancreatic disease. That to me is the power of deep learning, right? To be able to augment human insight by pointing to areas that may have been in our blind spot. And so this examination revealed that the cells that are most relevant to the different grades of tumor are actually the cells at the periphery of the tumor. And why is that important? If you think about it like an apple, if the core of the tumor, the seeds are the stem cells and the core of the tumor, and the white portion of the apple is the peritumoral region, that's a place that is actively growing. And that's a place of very high activity, which is essentially a war zone between the host tissue trying to fight the tumor and the tumor tissue trying to invade the host. So what this deep learning algorithm tells you, which is obvious to any pathologist, but was not obvious to me as a machine learning person, is that the cells in the periphery of the tumor are perhaps the most important when it comes to diagnosing a tumor or treating the tumor. And that has implications for immunotherapy. All right. So the last but not the least, again, to belabor the point, if you're in computer science and you've been doing geographic information systems or spatial biology, spatial analysis for other kinds of data, I would welcome you to think about biology, because this kind of data is now becoming extremely prevalent, and we're trying to still make sense of these aspects. So a third modality which has become very popular is called spatial transcriptomics. In spatial transcriptomics, you can take a piece of tissue, this is actually a piece of brain tissue, and you can obtain the gene expression for every single cell across the entire region of space. This is amazing. You can get a very deep look on the cartography of the tumor with a lot of detail. So again, there are tools to do this, and you can learn spatial signals that can discriminate the biology of the disease. So in the last few minutes, I'll tell you a little bit about a paradigm that we invented, which is my former postdoc, Santoshi Krishnan, who now works in a pharma company. And the question that we were trying to ask was, is there a way to be able to meld single cell non-spatial data with spatial non-single cell data, right? And can you combine the two to be able to obtain a object that you can do machine learning on that combines the best of both worlds? So we created this kind of a graph, which we call the proximogram. And the proximogram does something very simple. Every node of this graph is one cell type, right? And the node can have values that come from the single cell data, which indicate what is the identity of that cell type. So that's on the node level. The edges between any two nodes indicate their spatial proximity, which can be derived from spatial profiling platforms. So now you have a single abstraction that can bring both single cell non-spatial data and spatial data into a single object. And now what do you have? For every patient, you have a graph. So instead of doing classification on images, you're doing classification on graphs. Again, this is not a new idea from deep learning. People have been doing graph-based deep learning for a while now. But to our knowledge, nobody had an abstraction that could pull these two different modalities and create a single abstraction for it. So that's something else that we've done. So what my lab does overall is puts different kinds of modalities together to be able to make sense of it. And one thing we've learned the hard way is that QA/QC is so important. There are so many different modalities, so many different platforms that you require ways to be able to sensibly audit them. And this has been further challenged by the rise of agents like ChatGPT, which can do bioinformatics analysis. In fact, there are agents out there where you can just feed in a gene expression file and it'll do analysis for you. So while that's extremely amazing and you can do really amazing things with these kinds of agents, I guess the first question we need to ask is, how reliable are these things? So I like to always give this example to my students, which is um, I asked the question to ChatGPT, how many S's in strawberries? And it says there are three S's in strawberries. Just to be clear, there are actually two, but it says three. Then I asked the question, how many R's in strawberries? And it says three, which is correct. Then I said, you said there are three S's in strawberries, explain why. I mean, it gives me great logic, of course, wrong logic, why there are three S's in strawberries. It still doesn't pick up on the fact that it's made a mistake. Okay? So, then I asked, are you sure? Then it does a re-analysis and comes back and says, actually, you are right. There's only two. Then I asked the question, well, your ChatGPT is supposed to be doing really smart things. How did you make such a basic mistake? It makes up some excuse and basically tells me I took a mental shortcut. Right? Imagine this. A ChatGPT is telling me it took a mental shortcut. Right? So I said, well, you said you took a mental shortcut. Are you a mind? And it says, no, I'm not really a mind in the way humans have minds. I'm just a pattern recognizer. Right? So I use this example to have a lot of students who come to me and say, can I use ChatGPT to do my homework? I don't want to say no, because guess what happens when you tell kids to not use ChatGPT for their homework? Right? They will use it anyway. So you're better off trying to explain to them that these things come at a cost and that these things you need to verify. Right? And when it can fail in such a simple task of counting the number of letters in a word, you're expecting it to find genomic alterations to cure cancer. That sounds a little bit, out of the league of these instruments right now. I'm not saying they'll not get there, but it'll take a lot of cohesion in this community to be able to build tools that are actually helpful. Okay? I'm seeing a lot of interesting comments. I will definitely try to address them towards the end. But what I'm trying to tell you is that while this is definitely a way of the future, I think for mission critical applications like healthcare, cancer diagnostics, one has to be very careful what's happening under the hood. Otherwise, you get systems that are basically untrustworthy. And what I want to leave you with is that while there is hope for this sort of paradigm, I think there will be a real need for collaboration and domain transfer between clinicians, scientists, bioinformaticians, AI experts, all of whom need to speak a common language. And the only way to be able to get to a common language is through AI education and AI literacy. So I'm a big believer in, courses that can help educate the entire spectrum of individuals, from the AI deployers to the AI developers, to be able to help everybody understand what questions to ask of the AI before you can use it responsibly in your workflow. One more thing that we should probably think about is this is just the beginning of the story, right? We are literally in 2025 June, the middle of the year, and lots of things have already happened, right? We are seeing situations where ChatGPT first gets really good, then it gets really bad. Very, very unpredictable. The model keeps changing every time the data changes, right? The inferential guarantees, they change as the data changes. The rigor, reproducibility that we associate with science, that itself is being compromised. Things like transparency, trust, reliability are all becoming difficult to calibrate. So I think this is a time for folks from system engineering, software perspective, systems to come together and say, all this stuff is not new. Computer scientists, systems engineers, civil engineers have been doing this for a long, long time. But we need to bring that sort of thinking back to AI to say, how do we build systems that don't have any emergent failure modes and might not lead us to a wrong, turn. And like I'm saying, I'm not talking about what's going to happen next year or 10 years from now. I'm talking what's happening right now. Anybody, including you and me, can basically go to the web, pick up agents from different companies and put them together and create a complete workflow. That's not the hard part anymore. But the problem is that these companies have trained their agents on proprietary data with no cross-modal standardization, And so you don't know that when you are plugging in the output of Johnson and Johnson's API to Abbott's API is the transfer of state. The concept of state from a computer science perspective is the state properly calibrated and transferred from agent one to agent two. To give you a much more stark example, I'm showing you this cockpit of a Cessna biplane. It's a simple plane. It's not a very, very big plane. And these are all the dials that you see on this plane are the different sensors. One is an altimeter, one is a barometer, one is for all these kinds of different quantities. And if I told you each sensor comes from a different company and the companies have never talked to each other ever before in their lives, and all the heroes came and put everything into a cockpit one fine day, are you going to fly in this plane? Right? I like adventure as much as anybody else, but this is not an adventure I am willing to get on, right? Because if these companies have never talked to each other and there's no cross calibration of these sensors, God alone knows what the emergent failure mode is going to be. So when you won't do this with a plane, how are you so comfortable about applying this to healthcare? Right? These companies don't talk to each other. There's no standardization. Each one's doing their own thing in a silo. And standardization is still very, very early days. So until we're able to get there, I worry that these multi-agentic workflows are simply going to be proofs of concept and hobbies, right? For them to graduate from a hobby to something serious, I think that decision and that discussion needs to happen. And so the same thing applies when you talk about AI co-scientists, right? Google's got one, Sakara's got one, Gemini's got one, everybody's got an AI co-scientist. But think, science progresses when we do experiments that reduce uncertainty about a phenomenon. So the first thing you need to articulate is you need to articulate how uncertain something is to happen and then design an experiment to be able to reduce that uncertainty. But guess what? No AI system at this point, that's commercially available at least, ever reports its own uncertainty. What do I mean by that? You all use ChatGPT. I'm not pulling on ChatGPT. It could be Claude, it could be Gemini, it could be whatever you want. But isn't it curious, you think? It's been 2.5 years since ChatGPT came out. Don't you think it's curious that at no point is there even a color-coded response that says if the answer is in red, it's highly uncertain. If the answer is in yellow, it's somewhat uncertain. If it's in green, then the system is very certain. I don't think that's hard to design. Yet, at this point, all the output that comes out of a ChatGPT-like system is it looks like everything is equally uncertain or equally certain. And this is where the problem happens with going with AI agents for science. So I think these are nuances that I think we need to think about as a community. Otherwise, we're simply, kind of fooling ourselves. So, and this becomes even bigger, right? Is when you couple these AI agents to lab automation systems, you now have an amazing infrastructure at your hands. People are talking about a situation where I can just go sip Coke and, or Campa Cola and walk away, get a chai, and this experimental engine is going to design experiments, talk to a robotic platform and get data, and they're going to talk between each other and get me the answer to my hypothesis. That's the vision people are selling right now. But I wonder how we can even get there without this level of credentialing or calibration. So, it's very easy to dream up these scenarios, but I think this is a time for systems engineers from different communities to come together and truly calibrate these aspects if this has to become a reality. Otherwise, like I said, it's a nice proof of concept, but it will never actually make it to reality. Or people might still do it, and then after billions of dollars of spending, they might realize it was doomed to failure. So one thing I want to add as a part of bringing all this together is I think I'm a big believer in checklists. So there's a beautiful book by Atul Gawande called The Checklist Manifesto, where he talks about this fact that just like airline pilots have a checklist that they go through before they decide to taxi on the runway, They do a very, very clear systems check for every ingredient before they even, and all systems are switched on one by one in a very predictable order. We need to bring the same kind of thinking, I believe, to these multi-agentic workflows, right? And so we produced some AI readiness questionnaires and AI readiness checklists that are intended to serve the community from a couple of different perspectives. One perspective is if you're a company that's looking, let me take you to the, is this, no, let me, if you're a company that is looking to use AI and you put out a tender or a request for proposals from different vendors, and all these vendors respond to you and say, we can help you solve your problem. And everybody has an AUC of 0.99. How would you systematically assess the strengths and weaknesses of these different AI vendors before deciding who was right for you? But everybody's marketing material looks nice. Everybody's got glossy graphics. Everybody's got a 0.99 AUC, right? What will it take to be discriminating about these different vendors so you can make the best decision? So one checklist I'm happy to talk about is vendor credentialing. The other that a lot of companies think about is buy versus bid. Why would you, I mean, some people say, why should I buy something? I'll just hire a software engineering team and build it in-house. That could work, but again, that requires some due diligence. So we have questionnaires for that as well and checklists for that. So if you're interested, we could discuss that a little bit as well. But when I'm converging this to this notion that slowly but steadily, guidelines and checklists are coming online that will let us establish the veracity of these different tools. And if as a community we can agree on what elements make sense for our individual situations, hopefully we'll see a lot more systematic success. Otherwise, it'll look very sporadic and there'll really be no method to this madness. Like I said, one of the biggest things that I want to get into, so if you're interested, please reach out to me, is I'm very interested in corporate education. I'm very interested in aspects of educating the continuum from the AI engineers all the way to the AI deployers, which could be the chief scientific officer, the chief operating officer, the strategy officer, whatever it is, right? Because I think as only when everybody in this continuum is educated about the potency of AI, can we actually hope that AI will get leveraged responsibly, ethically, and in a pragmatic fashion. Otherwise, we are probably selling snake oil, and we want to be careful about not prematurely creating an AI winter. So this is the kind of course I do in my thing. If you're interested, please reach out to me. I'm happy to talk about that. It's basically meant for AI in radiology and pathology, but the principles are general, and I don't talk about math. I'm talking about building intuition as a way to be able to understand and ask the right questions. So I'll close by firstly thanking the people who made the first project possible. Now it was the clinician that gave us the problem. Jeff and Justin helped generate the data. My students, Rachel and Priyanka, did all the work. So I'm really, really grateful for the opportunity to work on this with them. The immunology stuff, is a multi-team effort. I'm an engineer by training. I don't come from biology, but a variety of people helped us get here. So with that, I'll close and I'll say, if you have any questions, please feel free to email me or interact further. I would love to hear from you. Again, thank you for your attention and really, really apologize for the first 10 minutes trying to get this whole thing set up. But happy to take questions. Thank you.
Yeah, wonderful, Arvind. You spoke very lucid biology. I mean, it never sounded that you are not a biologist. Thank you so much, for a wonderful talk, Arvind. Yeah, yeah. So I have few questions, but as a co-host, I'll probably reserve my questions later. But those of you, please please switch on your camera and raise your hand and ask Arvind, unmute your microphone and please introduce yourselves very quickly. And please ensure that you give probably short and sweet questions so that there could be room for others. Sim, please.
Sir, thank you for giving me this opportunity. It was a very nice lecture. You have offered even PG student fellowships and postdoctoral fellowships in your lab. That's great. We will send our students to you. And we like to collaborate with you. If you are having the opportunity to collaborate with Indian scientists, especially like me, you please offer us.
Absolutely. Please send me an email. I would love to take this conversation forward. Thank you.
Thank you, sir. Thank you.
Yeah. Anyone else, please? Yeah. Yeah, there were some kind of discussions that Bhupen and Manjari were discussing. So Bhupen and
Yes.
Manjari, would you like to ask any questions, please? Yeah. Yeah. So while others ask, maybe I'll just take this opportunity. I was just wondering, Arvind, how tangible or how feasible it is to consider, mesenchymal cells, as a part of your this thing. I know, we could even give a kind of a score, for mesenchymal cells, based on the range, based on the perturbations that they could invite or whatever. So could you look into those things, Arvind? Yeah.
Oh, absolutely. So I think what we talked about was based purely on morphology, but with the advent of sequencing now becoming so much more affordable, like you're saying, there are mesenchymal signatures that we can use to flag the extent of mesenchymality, if I could use that word,
Right.
right? And then now it's even better because you don't have just a binary characterization of epithelial versus mesenchymal. You can actually have an EMT score and use that as a way for regression, which is even better because now you get a much better handle on those uh, perturbations that might affect the grade of mesenchymality. So
Right.
that's,
Okay.
yeah.
So having said this, since it's a multi-omic approach, I know you spoke a large entity about the codex, which is largely for proteomics, spatial
Yeah.
proteomics. But
Yes.
there are other suites like Cosmex, which is largely used for spatial transcriptomics or
Yes.
Visium. So all the well-known seven spatial omics technology technologies could probably be inferred. But when you said multi-omics, from your first experiment which you showed, isn't genomic or exomic technologies missing from that? I could see only the transcriptomic and probably proteomic
Oh,
things.
right, right. So, yeah, it was not meant to be exhaustive. In fact, one thing that was very, very noticeably missing was also epigenomics, right?
Right.
And so now there are technologies for spatial epigenomic interrogation in addition to bulk. So I think the aspect that I was just trying to insinuate was that given the multi-omic nature of biology, and the fact that each modality has to be analyzed in its own special way, there's, I think, a great opportunity for statistically informed approaches to bring all these modalities together, leveraging the physics that we have knowledge about.
Right. Wonderful, Arvind.
That's
Sugu
all it's
has a
trying.
question. Sugu is a senior scientist at Indo-American Cancer Center in Hyderabad. Sugu, please. Yeah.
Yeah, hi. Thank you, Prash. There was a great talk, Dr.
Thank
Arvind.
you.
I was trying to look into. You talked about something called zooming into the cells, the electrogram pictures, where you talk about noisy kind of things, right? So, how do you do a kind of normalization there?
Uh, so when you talked about, you mean noise in the instrument, or did you mean batch effects between instruments?
Batch effects within between instruments. Yeah.
Yeah, so I think the key part, I think what we did was we used a algorithm called Combat, which is used for batch effect assessments, even for gene expression data. So we used that as our way to clean up or account for that variability. And then after you subtract that signal, what you are left with is what we analyzed for insight.
Okay. And from a point of the cells, you talked about individual wells. 96 wells that you have it, and you zoomed in, and then you did the kind of
Pictures,
projections.
yeah.
Yeah, pictures.
Yeah.
And how did you even normalize that?
So what we had was, because we had positive and negative controls on the left, the ones that were
Yes.
multicolored wells. So they gave us a range of what these cells could do under different um, environments, and we used that as a way to do like a max min normalization. So we took every intensity, calibrated it in the same intensity range, and then got all our features based on that. Which is exactly, it's a great question because that's exactly why I focused on morphology and not on intensity. right? Morphology features will persist even with variable intensities. But intensity, if you only take those features, they can be affected by these batch effects, which is why we didn't take intensity features.
Okay. Okay. Thank you. I'll connect with you, Arvind. Thank you.
Absolutely.
Yep. Yep.
Absolutely.
Thank you. Thank you, Prash.
Yeah, thank you. So, assuming that there are no more questions, I just probably have one more question, Arvind. I'll just take liberty. I was just wondering, in case of breast cancers or in case of cancers, there are something like radical cancer tumor types as well. So when we visualize or spatially transcriptomize the data, would we be willing to would we be able to fetch those specific records or genes per se that are differentially expressed between between those radical cancer tumor types? I mean, could that be a proposition that can be taken?
Absolutely.
Yeah.
Very much so. In fact, I skipped over that in interest of time, but TNBC has heterogeneity. There are about, I think, 6 subtypes to TNBC, ranging from basal to luminal, those kinds of things. And so one can use those gene signatures to now find subtype specific perturbations that might be capable of reversing the extent of the aggressiveness. That's definitely
Right,
feasible.
and that way, in fact, bulk RNA seq need not be a norm, right? I mean, we can step directly into a single cell technology, and
Absolutely.
that's where I mean, as a sequence biologist, I would always argue that we need bulk RNA seq first, to understand those segregated differential expressed genes, and then zoom into certain specific cell types, to understand the differential expressed genes. Yeah, thank
Yeah,
you.
I couldn't agree more. I think, in the interest of cost and scalability, bulk is definitely my favorite choice as well. And so I think of both the single cell and the spatial modalities as ways to fine tune the um, ability to call those subtle cell types from the bulk data through
Right.
deconvolution. So I feel it's very important to recognize that don't throw away the old data in trying to pursue the new kind of data. There's a lot of value in trying to keep these things mixed across these various modalities.
Right. A couple of questions from Imran. Imran is a
Yes.
postdoc here. Postdoc. Yeah. Imran, please quickly. Yeah.
Thank you, sir. Yeah, first of all, sir, it was a very lucid talk. So
Thank
I
you.
just had only one question. Like you mentioned, proximogram, if I'm pronouncing it
That's right.
correctly.
Yes.
Yeah, in terms of the spatial biology interpretation that you did. So is it the only way to interpret the spatial transcriptomics data, or are there any other graphical representation by which we can do this analysis?
Oh, I think there are plenty of ways. The reason I was talking about the proximogram was it's the only way that I know where you can bring two disjoint modalities together onto the same structure, right? So, like I said, what if you had single cell data from pancreatic disease and you had spatial data from pancreatic disease, but not on the same specimens, right? So, if you have matched data, then you can do whatever the existing tools do. What we tried to ask was, what if your data is not matched? So if you had, as long as the disease cohort is the same, you can use that to be able to look at these kinds of things. So that was the whole point of the proximogram.
Okay, thank you. Thank you so much, sir. That's all from my
Absolutely.
side.
Yeah.
Absolutely. Thank you.
Yeah. Thank you, Indrani. Bopen, please. Yeah. Yeah. Bopen Singh. Yeah. Please. Can you please unmute your microphone? Yeah. I think he left accidentally. I don't know. Yeah. I don't see him. Yeah. But he raised his hand. I don't know.
Yeah, I think I saw that too. Maybe he dropped off. Yeah.
Yeah. Yeah. So with this, I think I end my case. But over to HR Mohan sir for for his vote of thanks, please. Thank you.
Yeah, good evening. Thank you very much, Dr. Arvind, for your
Thank you, sir.
wonderful presentation on AI for biospatial spatial biology analysis. Thanks for your presentation based on image informatics, and also particularly highlighting how you can interpret data with a relatively smaller data set, and then predicting with a larger amount of data. So it is going to be a sort of a really a tough job for the scientists. And thanks for stressing the need for AI literacy among all the stakeholders, in this research, and particularly, with the chat GPT and things like that, creating a lot of confusion. And of course, you really highlighted the limitations of chat GPT, and then the people's interest in using for various purposes. And you have really pointed out about verifying I mean, inferences by the chat GPT. That is a real crux of the use of chat GPT. And definitely, when things can go wrong in even the textual matter, particularly in biological and life sciences data, it's going to be extremely difficult to find out because no one knows the answer. At least, some people know the historical information. So, on a textual matter, you can easily find out. And another interesting thing is you have really highlighted the AI washing in multi-agent workflows. In fact, everyone is doing the AI washing. Suppose, if you open the newspaper or any magazine, there is over emphasis on, how the AI could solve the problem, something like that. I mean, as you pointed out, it is extremely difficult to do the science with the interpretation. So actually, the experimentation is so. Though in fact, in the early days, bioinformatics itself, people are not believing, right? So basically, the wet labs, or was very much. In fact, bioinformatics people are feeling left out. Their predictions, are not given enough importance and things like that. Now, at least to some extent, it has become one of the most accepted field. But with the AI coming and trying to infuse AI in research, in fact, we need to really look at what could be the outcome, and we have to keep our fingers crossed. So,
Sure.
thank you once again for your excellent presentation, and
Thank
thanks
you very
all
much.
the participants who have joined this webinar. And we look forward to have many such webinars trying to bring computer science and biologists together in a sort of this forum. And Dr. Arvind is in India for I think few more weeks he is going to be there, and he is also expressed his interest in providing webinars and as well as meet the people. And in fact, on next week we are going to have almost a week presentation in Chennai. So he is also expressed his interest in visiting few places. And Bangalore is another place where we are going to have few more talks, including IBAB, now they are planning. And at Chennai, IIT Madras, and Institute of Mathematical Sciences, and Anna University Center for Biotechnology, and Cancer Institute, Adaya, and VIT Vellore. So these are places the Chennai visit will cover. And definitely, some of you are interested. You can always get in touch with Dr. Arvind, and we can make use of his visit to our best. Thank you very much, Arvind.
Thank you very much for
Yeah,
your warm
before,
welcome
yeah,
and for
It is my sincere thanks to Prasant for coordinating this entire. I think
Thank
we
you.
just made a request, and then Prasant took the lead, and then
yeah, no, it all, sir. Thank you so
everything.
much for your
Thank
this thing. Can we
Thanks.
all have a group photograph, please? Can we all have a
you.
group photograph? Yeah, there are few cameras that are coming up. Yeah, thank you so much, everyone. Um, yeah, Ashish, Venkat, Chaitra, um, Kishore, and Kausanayas. Yeah. Otherwise, I will quickly take this photograph. But as they switch on their camera, I'm always very much disturbed with this note takers, Arvind. In this Google this thing. I always wonder these AI based note takers. They record something, and probably they misinterpret as well. And I don't know what would be the predictions. So maybe इसके लिए कुछ इलाज करना चाहिए. I think there must be some remedy for this. You know.
Yes.
Mm, yeah, I'm
No,
just
I
taking.
think the way is to make it to make these agents be adversarial. So what, for example, they say is take ChatGPT output, feed it to Claude, and say that's what ChatGPT said. What do you think? So now we create a competition, and then that's how these things become better. Maybe that's the way.
Right. Thank you so much, Arvind. And Thank you, everyone. In fact, Arvind, if you just have 2 minutes, there are several other people here, whom I just wanted to introduce. We have,
Sure.
Professor Nirmal Loya sir, who is our
Hello.
advisor and mentor as well, just like HR Mohan sir. He is a distinguished emeritus medical scientist in University of Rajasthan. We have Ansari sir as well, who is a professor here in University of Rajasthan. And we have Sri Manjari here, who is our secretary. Subhu is already introduced. Yasin, she is a senior scientist at ICAR. She would probably mail you. We have several other people. Subhu, of course, asked, and we have Akshay from Gene Spectrum Life Sciences. He's a founder director over there. We have Sudeshna Nag, Himani Sharma, and other people from, and Gopesh, of course, from Jaipur, but Gopesh is currently in US. Chaitra Murthy, she is from Bangalore. Kausanayas is a founder of Abir Bioservices based in Delhi. And we have few others like Ashish, who is a senior scientist at CSR NEAST. We have, of course, Dr. Sakila Ganesh and Komati from Mohan Sir's initiative from IEEE. Of course, Kishore Shinde is a senior principal scientist. He's a well-known fungal biologist. And we have others. Sohan Banerjee is a PhD fellow based in Academica Sinica in Taiwan. And Pragati Gupta and others, I apologize, that's what. So I just thought
That's wonderful.
introduce, and this talk is being recorded, and I'll probably put it up in YouTube page of Bioclues in moments from now. Thank you so much, friends. Let's put together our hands. Please unmute your microphone so that Arvind hears it all the way to the States when he goes back.
That's the kind of you. Thank you.
Thank you.
Yeah. And of
Thank
course,
you,
Varun is there. Varun is also.
Thank you.
Thank you so much.
Thank you. Bye. Thank you, everyone.
Thank you, Arvind sir.
Thank you.
Thank you. No problem. Bye.
Thank you, sir.